Hi, my name is Dresden Feitzinger.
I'm Joe Urbano.
We are here to talk to you about detecting a certain type of white blood cell in a nasal biopsy sample called an eosinophil. So many people suffer from a disease called chronic sinusitis, and it eventually sometimes leads to surgery, and the doctors must determine how to treat the patient after the surgery. So they call in pathologists to provide a qualitative measure of the eosinophils in a nasal biopsy sample from the patient. And increasingly, instead of a qualitative measure, the doctors are looking for a quantitative measure. And in fact, they're looking for an exact count so they can provide better treatment for these patients. So our original approach for this was to take a whole slide image of these nasal biopsy samples, segment it to obtain the individual cells, perform binary classification on each of those cells, and then count the number of those cells which the CNN believes is an eosinophil with certainty above a predetermined threshold. Joe and I wanted to see if we could extend this approach to achieve better performance. So our new approach, we create a probability map. We use a sliding window protocol to basically cycle through a whole slide image and feed each tile image to the CNN and create a probability map using the output of that CNN. We then use an algorithm called local maxima detection to find the modes of the probability map, basically the peaks in the probability map, which hopefully correspond to the eosinophil cells. So this process of feeding all the tiles through the CNN, there's thousands of these windows, can be very time consuming. So we decided to try and integrate decimation into our system, where instead of taking every pixel for every window, we might take every n pixels for a window. And in fact, we found that this has little effect on the overall result, but does achieve significantly better performance. And if you notice the curve on top of the slide here, you'll see that with a decimation factor of 1, meaning we're taking every pixel, it takes over a minute to complete this entire process. Whereas with a decimation factor of 10 or 15, the runtime is almost negligible. And note down at the bottom, with a step size of 10, we can see that decimation captures the overall structure of the probability map sufficiently well. And it also has the added benefit of reducing noise in the probability map.
Alright, so let's jump right into the results here. So there are two things that we were looking to measure. That's the runtime and the accuracy. And we wanted to compare our new approach to the old approach. So what we have here is a plot of the running time versus the tile size. So as the tile size increases, the running time obviously increases as well. And we're using a step size of 10 as a decimation factor for our probability map. So as you can see from the data here, the results are pretty comparable to our old approach. The probability map approach is a little bit slower, but not significantly slow. But overall, the approach of segmenting and then classifying does win out. All right, so we'll take a look at the accuracy here. So we have a data set of tiles that was labeled by a pathologist at the University of Pennsylvania. There's a total of 408 tiles with a total number of eosinophils of 921. And we measured for both of our approaches, we measured the recall and the precision to try and gauge how accurate our system was. We can see that our old approach of first segmenting and then classifying has a higher recall of 12%, while the probability map approach has about only 2/3 of that. So that's a pretty significant drawback to the probability map approach. And we'll kind of see why on the next slide. There's a limitation that I talk about. But the precision of both approaches is pretty comparable. So precision is, of the things that we classify as eosinophil, how many of those were actually right? So I mentioned the limitation of the probability map approach, and that comes into play when there are a lot of eosinophils that are in close proximity of one another. Because what ends up happening with the probability map is you just get a big cluster of high probability and just gets all lumped together. And so when you find the local maximum of that, you just get 1 point, and there's no distinction between where the individual cells are within that blob of high probability. But this is somewhere where the old approach of segmenting and then classifying really excels, because when you segment out the nuclei, you really know where the individual cells are. So that kind of really gives a bonus to our old approach. So overall, the final verdict we would say is the new approach, really doesn't work as well. We kind of stick with our old approach. But some future work for this, we can definitely improve the CNN. One thing that we can improve on is, the data set that we use to train it. We have a relatively small data set. We only have a few thousand images of eosinophils to train on, and it's arguably not enough to get a really good classifier. And on top of that, we have a pretty simple CNN. It's only a 7-layer network, and we designed it to be thin so it'd be fast to classify a lot of images. But the accuracy is limited, so it might be better to maybe do transfer learning on a better network such as ResNet or something like that. And on top of that, we can look at other means of downsampling the image, other than decimation, maybe something that might give slightly better results. For example, superpixels comes to mind. That's something that we wanted to try, but we didn't get around to. So that's all. I hope you enjoyed it. These are the references. Thanks for watching.